Hello and welcome to Sam Biology class. Friends, today we shall discuss about cloning vectors. Cloning refers to making identical or virtually identical copies of an organism, a cell or a DNA sequence. Molecular cloning is isolating and copying a particular DNA segment of interest for further study. What is vector? A vector is a DNA molecule used for carrying an exogenous DNA into the host organism and facilitate its stable integration and replication inside the host system. The vector which is used for cloning is called as cloning vector. So cloning vectors are useful for making very large number of copies of a particular DNA segment for further study. A suitable cloning vector should possess the following features. First one is origin of replication. This is the sequence which is essential for replication of a DNA segment. The presence of origin of replication in a cloning vector enables its autonomous replication inside the host. Second feature is restriction site. The specific site at which the restriction endonuclease cleave the DNA is called as restriction site. The presence of restriction site in a vector enables insertion of a foreign DNA segment into it. Third one is a suitable cloning vector should possess a selectable marker that will enable the screening of recombinant organisms. And fourth one is the cloning vector should be small in size so that it can be easily integrated into the host system. And further, the cloning vector should be capable of inserting a large segment of DNA. And finally, the cloning vector should be capable of working in both the prokaryotic and eukaryotic system. Vectors can be divided into two different groups. Number one, cloning vector, and number two, expression vector. Now we shall discuss about some of the cloning vectors which are commonly used in genetic engineering. First one is plasmids. Plasmids are extrachromosomal autonomously replicating DNA molecules which are found naturally in bacteria, archaea and few eukaryotes. Most of the plasmids are circular double-stranded DNA molecules. They have an origin of replication, so they are capable of autonomously replicating. Plasmids can be divided into two different groups. Number one, natural plasmids, example COL-E1, RSF1010 and R100. In addition to natural plasmids, artificial plasmids are constructed in vitro by recombining different segments of a plasmid. Example, PBr322, PUC18, PUC19 and so on. Artificial plasmids are considered efficient than natural plasmids because they have additional features. In addition to plasmids, certain bacteriophages such as lambda phage and M13 phage are also used as cloning vector in genetic engineering. Lambda phage and M13 phages are considered more efficient than plasmids because they can accommodate a very large DNA segment up to 53 kb. There is another unique group of cloning vectors called as phasmids, which are DNA based. They have the properties of both plasmid and the bacteriophage. In addition to its plasmid origin of replication, they have an additional origin of replication derived from bacteriophage. Unlike plasmids, phasmids can be packaged into capsids because they have the specific sequence that signal for packaging. In addition to this, there are other cloning vectors such as bacterial artificial chromosome, plasmids, yeast artificial chromosome, and human artificial chromosome. So friends, this is all about cloning vectors. In next video, we shall discuss about expression vectors. For more updates, please subscribe my YouTube channel Sam Biology Class and please share your comments. Thank you very much.